Hello, I'm Luna Spears, and I'm going to talk about how we can isolate mRNA using magnetic isolation. We're going to go through the introduction, methods that we use, and applications for magnetic isolation of mRNA. Let's introduce mRNA. mRNA is a molecule that encodes all of the proteins in our body using the DNA in our nucleus through a process called translation. This mRNA brings the information to the ribosome for protein formation. We only make it when needed, and as eukaryotes have post-translational modifications that we undergo in order to make mature mRNA. This includes making a 5' cap, removing intronic regions, and making a poly A tail. We use mRNA in the COVID vaccine to make proteins we need to fight the virus. Now let's talk about the process of adding a poly A tail. In pre-mRNA, there is a tail recognition sequence as seen in the slide where endonuclease cuts after CA to expose the 3' hydroxyl group. Polyadenylate polymerase adds a stretch of adenines between 70 and 200 base pairs depending on the species in order to increase stability of mRNA, protect from degradation, and aids in transport outside of the nucleus. We can use this poly A tail to isolate mRNA with a magnetic isolation. This is important because of RNA sequencing, where mRNA only contains exons after the introns are spliced out, and exonic regions are important for gene expression. It tells us what genes are being expressed at that moment based on alternative splicing or different patterns over time in different conditions. This was not invented by one person, but is an accumulation of knowledge and lots of trials to become effective for RNA isolation. There are lots of different methods used, so let's talk about some of them. Before we can isolate just mRNA to have higher success, we need to isolate the total RNA from our sample. There are many methods for isolation of RNA, including phenol extraction, which is the most commonly used, which has rapid denaturation of nuclease and a stable RNA, but is difficult to automate and is manually intensive. We can use filter-based membranes, which are convenient and able to automate, and we can manufacture many membranes, but it can clog with particulate, retain large nucleic acid like gDNA, and has a fixed binding capacity. Magnetic particle has no clog risk and has rapid collection, efficiently captures the target RNA, but can carry magnetic particles into samples and slow migration in viscous solutions. We can use direct lysis, which is fast and easy, has a high potential for accurate RNA, and works with small samples, but we can't perform any other analytical methods because it's dilution-based and has potential for unwanted residues. Because organic phenol extraction is considered the gold standard of RNA preparation out of our method options, we're going to talk about it today. It yields the best results for mRNA extractions later on. First, the sample is homogenized in a phenol-containing solution. Second, the sample is centrifuged, separating into 3 parts, as seen in this image: a lower organic phase, a middle denatured protein and DNA phase, and an RNA aqueous phase. The aqueous phase is what we collect. In step 3, that RNA is precipitated out using sodium acetate and ethanol. Step 4, the sample is centrifuged, separating the RNA nucleic acids into a pellet, which is washed with ethanol to remove any residual salts. In step 5, the nucleic acid pellet is resuspended in a clean aqueous buffer for analysis. Now let's talk about separating mRNA from the total RNA. mRNA only makes up 1 to 5% of our RNA. It tells us what proteins need to be made in our bodies in that exact moment. We use oligo-DT coupled with magnetic beads to isolate just the mRNA because thymine binds complementarily to the adenine tail and the magnetic beads allow easy separation. We use kits like Dynabeads, PolyAtract, or NebNext as some examples, including a lot more, all of which use oligo-DT primers and paramagnetic beads. We do not need to precipitate it, and it's a quick process. Because these kits use the same process, many of their protocols are the same. I am looking at the PolyAtract and its protocol of large-scale PolyAtract systems. First, oligo-DT probe and buffer are added to a separated and heated total RNA. This is mixed and incubated to room temperatures to allow for annealing to the poly A tail. Next, compressed paramagnetic particles are resuspended, separated with a magnetic stand, and washed with the buffer. Third, annealed solution and paramagnetic particles are mixed, intubated, and separated with the magnetic stand to pellet and capture the oligo-DT mRNA hybrid by removing the supernatant. Particles are washed, and the final pellet is resuspended with RNase-free water. mRNA is magnetically captured and transferred to a new tube for an analytical analysis. Now let's look at some applications of use of mRNA isolation. In this study, Matkovich and colleagues sequenced cardiac microRNA and mRNA interactomes in order to study cardiomyopathy. He used an experimental approach of pulling down mRNA associated with the RISC or RNA-induced silencing complex protein with immunoprecipitation to avoid biased capture. He measured mRNAs, microRNAs, and RISC-bound mRNAs using the poly-A magnetic isolation that we saw. He then sequenced using Illumina. This study showed how microRNAs regulate mRNAs in diseased hearts. We see which mRNAs undergo regulation. This regulation occurs with direct microRNA-mRNA interactions that are critical to heart function. By identifying which mRNAs are influenced by RISC, we can explore therapeutic options. Fick and colleagues studied another important approach of mRNA through defining the 5' and 3' untranslated region sequences in our favorite fruit flies, T. melanogaster. By mapping transcription start sites in the 5' end and understanding the 3' end, we have a better understanding of cell physiology. By refining our genome annotations and identifying different isoforms across the same species, we have a much better understanding of the necessary regions mRNA ends. The isoforms are all determined by mRNA with different splicing and important regions. The scientists also explored the effect temperature has on these regions and showed that temperature fluctuations change the exact boundaries of the mRNA. This influenced which 5' start sites were used and therefore influenced gene expression. It also showed new CA cleavage in the 3' end as well as differences in polyadenylation. In this study, Scholes and colleagues went back and compared different methods of RNA isolation to prove which one had the best results for mRNA isolation. This allowed the analysis of differential results based on the extraction methods. The scientists found that mRNA transcripts encoding membrane proteins had a much higher abundance in phenol extraction, which we used in this PowerPoint, suggesting that the chemistry of hot phenol better suits mRNA structure. They are strongly enriched for transmembrane transport, localization, and cell wall organization. All RNA isolation still yielded a high-quality mRNA, but phenol compared to different kits used in the study has yielded the highest abundance. Here are the many sources that I used, and thank you so much for listening.